Hello friends, in this video tutorial we'll be talking about the glycogen metabolism. Glycogen metabolism is very important because glycogen is a much more complex form of glucose, and glucose can be stored in muscle cells as well as in liver as glycogen. Now glycogen is stored when we are having a lot of amount of glucose in bloodstream, but when we are in a starved condition we need to produce glucose, and in those situations the rapid generation of glucose can easily be done by breaking down glycogen. Now here glycogen is a polymer of alpha 1 fold linked glucose with alpha 1,6 branches, as you can see in this picture, and this branching can be found in 1 in every 10 residues. Now liver uses glycogen as a reservoir for maintaining blood glucose level. Muscle uses glycogen as a reservoir of glucose for energy production, because muscle need to produce energy most of the time. Now glycogen is built from a single starting point involving attachment of the first sugar to small protein called glycogenin, and the glycogen is found as granules in the cytosol, as you can see here in this case. Now the major glycogen degradation enzyme is glycogen phosphorylase. It requires pyridoxal phosphate or PLP for activity and uses inorganic phosphate to split the 1,4-glycosidic bond and produce glucose 1-phosphate from this glycogen in each round when it degrades the glycogen. Okay, now this glycogen phosphorylase is having the cofactor of pyridoxal phosphate we have seen here. Now this glycogen phosphorylase is in non-hepatic tissues. Sugar released in trapped in the cells since it is phosphorylated. Now pyridoxal phosphate is a derivative of vitamin B6, which is pyridoxine. Now phosphorylase can actually only work until 4 units of the branch. So that is a disadvantage of this glycogen phosphorylase. This enzyme can only act from the non-reducing end of glucose, and second thing is that this can only attack and break alpha 1 fold linkage. It cannot break alpha 1,6 linkage. And the third important thing is that this phosphorylase activity will only work until 4 units from a branch of glycogen. Okay, now let us continue. Now the conversion of glucose 1-phosphate into glucose 6-phosphate is freely reversible and is also important in utilization of the glucose, because we need to produce an intermediate product which can be uptaken or which can be taken by the glycolytic pathway. So glycolytic pathway is having the intermediate which is called glucose 6-phosphate. It is not glucose 1-phosphate. So we need to convert glucose 1-phosphate into glucose 6-phosphate. It's just a change or transfer of phosphate group from one carbon position to the 6th carbon position, as you can see in this picture. And this conversion is mediated by the enzyme phosphoglucomutase, and the cofactor for this enzyme is 1,6-bis-phosphoglycerate. Now 1,6-BPG is very important in this case. Now the key point about this reaction is that this is similar in the mechanism to the phosphoglucomutase, which needs glucose 1,6-bisphosphate as a cofactor to keep the enzyme active and phosphorylated form. Okay. Now to degrade the branch points, 2 additional enzymes are required. One is the transferase, which switches residues near the branch point, and another one is the debranching enzyme, which hydrolyzes the 1,6 linkage and produces this free chain. We have just seen this before. Okay, now both of these enzyme activity of transferase and debranching enzyme activity is performed by the debranching enzyme. Now the glycogen is synthesized by the transfer of the glucose from UDP glucose to the end of the strand and glycogen granule. So we are having this glycogen N-1 and we are having UDP glucose. Now the glucose is transported, it is given to so it's a shuffling of glucose with glycogen. Now UDP is released. Now glucose is attached to the glycogen and it will incorporate with the glucose with alpha 1,4-glucosidic linkage. As a result, it will produce glycogen. With the help of the protein glycogenin, it will make this granular structure. It will help it to store into the cytosol of a cell, and the enzyme is required for the synthesis of glycogen is called the glycogen synthase. Now synthase works at the time of high blood sugar concentration, because if we are having high sugar, that means we need to store some amount of sugar for the future times. In those situations only the storage of glucose as a form of glycogen occurs. Now UDP glucose is formed from glucose 1 phosphate and UTP. Additional of 1 glucose to glycogen requires an ATP to produce glucose 1 phosphate. From glucose via glucose 6 phosphate it can produces it. So glucose 1 phosphate can be produced from glucose via the glucose 6 phosphate, and UTP as used in this step. So we are having this glucose 1 phosphate and we are having this UTP. Now what it does, 2 phosphate groups are released, so only UDP is there. UDP is attached to the glucose to produce UDP glucose. Now this UDP glucose formation from UTP and glucose is called the UDPG, is mediated by the enzyme called UDPG phosphorylase, because it is only about phosphorylation. Formation of the nucleotide sugar is driven by the phosphorylasis of the released PPI. So this the del G negative value gives rise to the enzyme due to the hydrolysis of the spirophosphate, which is released right after the attachment of UDP with glucose. Now branching occurs when branching enzyme breaks one strand and transfers the fragment to the 6 carbon of the glucose to another strand, as we have seen here. So we have a linear, we are having both of these linear sequences of glycogen. Now branching enzyme can come and cut a small part of the sequence here, and it will take and attach this part of the sequence with this another or the previously created part of the sequence via the alpha 1,6 linkage, and we call them the branching point of this glycogen synthesis. And the branching point synthesis is mediated by the branching enzyme, and similarly there must be debranching for the degradation of this glycogen, and the enzyme which is responsible for the debranching is called the debranching enzyme. And the key point about this point is that the branching allows faster degradation and synthesis, since there are more ends to work on at a time. That's why these branching are favored all the time, because it allows faster degradation and it also allows a faster synthesis. Okay. Now if we go to the control, the glycogen is simultaneously being formed and degraded. Complexes hormonal control determines whether there is a net formation or a degradation. Phosphorylase activity and the glycogen synthase activity are affected by both covalent modifications via the phosphorylation event. Now glycogen phosphorylase, when it is phosphorylated it becomes activated, but synthase, when it is phosphorylated, it becomes deactivated. Now activation of cyclic AMP dependent kinase or protein kinase A, or aka protein kinase A, leads to the phosphorylation of both this phosphorylase and glycogen synthase enzyme. Now cyclic AMP dependent kinase is activated through a heteromeric G protein, and we all know all the signaling of this G protein all the time. Now what happens, cyclic AMP dependent kinase phosphorylates and inhibits the glycogen synthase directly. Now when this glycogen synthesis is unphosphorylated, it is active, and when it is phosphorylated, it is the presence of this phosphorylase kinase, it is inactivated. However, in case of the phosphorylase enzyme, when it is phosphorylated it becomes active, when it is dephosphorylated it becomes inactive. Now you can see here higher concentration of calcium ion make this phosphorylase enzyme much more active, and even much more active for all the time. Now these are the two different types of, or two different forms of phosphorylase enzyme that are present. One is phosphorylase A, which is much more active, another one is phosphorylase B, which is less active. Now the phosphorylase phosphorylase B can be converted into phosphorylase A by attaching or by the presence of AMP, and also due to the presence of phosphorylase kinase enzymes. Okay, now the calmodulin in another protein, which is a calcium binding protein, is a subunit of a muscle phosphorylase kinase. When the sarcoplasmic reticulum releases calcium during the muscle contraction, the calmodulin subunit binds to the calcium and changes the phosphorylase kinase to an active form, and as a result of the production of this active form, it phosphorylates the glycogen phosphorylase. As a result, glycogen phosphorylase can phosphorylate glycogen units to produce glucose 1 phosphate. So that means it ultimately leads to the generation of glucose in much more amount. So this is the overall summary of the glucagon signals. So glucagon signals lower blood glucose level. The liver responds by degradation of glycogen and release glucose 1 phosphate. Then finally glucose 1 phosphate is converted into glucose 6 phosphate, and throughout the glycolysis pathway it will lead to produce the pyruvic acid. Now inside the muscle, and the epinephrine signals all the pathway. Epinephrine signals G protein through the beta receptors to degrade glycogen. Then the glycogen is degraded and production of glucose happens, and it will burn up all those glucose to yield the amount of energy. Now we were here about the glycogen synthesis in this point. Now let us move on. Now these are the steps. The glucose 6 phosphate is a glycolytic intermediate. Glucose 1 phosphate and UDP glucose are a part of the galactose pathway. So these are the linkage of all these things. Now thus the glycogen is an effective means of storing quickly available substrate for metabolism and energy, because we can easily convert this glucose into glycogen pretty fairly, and we can easily degrade glycogen into glucose when we need to produce energy. Okay, so that's it, and I hope it will help you to understand. Thank you.